Good afternoon and welcome to the IGEPI seminar series presented to you by the Center for Communicable Disease Dynamics and the Harvard T.H. Chan School of Public Health. My name is Benet and I'll be your moderator for this week. Today we're delighted to welcome Professor Iruka Okeke. She is a professor at the Faculty of Pharmacy at the University of Ibadan, Nigeria. Just a few logistics before we start. This seminar is being recorded, so please turn off your camera if you do not wish to be recorded. The recording will be available on our website at a later point in time and on YouTube as well. After approximately 45 minutes, we'll stop for questions. Throughout the talk, you can send your questions via the chat function to Flavia Componovo, who is moderating the chat today. You have the possibility to ask your question to Professor Okeke directly. In that case, please notify Flavia in the chat. We will ask you to turn on your camera and we will unmute you once we are ready to take your question. Coming back to our speaker, Professor Okeke is a molecular bacteriologist focused on diarrheal pathogens, bacterial drug resistance, and laboratory practice in Africa. Her laboratory studies the molecular epidemiology, pathogenesis, and genomics of enteric bacteria, and provides genomic support for public health surveillance of antimicrobial resistance in West Africa. The Okeke Lab also engages in collaborative research to develop new therapies against neglected enteric bacteria. Professor Okeke received a Bachelor of Pharmacy, Master of Science, and PhD degrees from Obafemi Awolowo University, Nigeria. She did her postdoctoral training at the University of Maryland, USA, and Uppsala Universitet, Sweden. She is the author, co-author of several scientific articles and chapters, as well as the books Divining Without Seeds, The Case for Strengthening Laboratory Medicine in Africa from Cornell University Press, and Genetics, Genes, Genomes, and Evolution from Oxford University Press. She is editor-in-chief of the African Journal of Laboratory Medicine. A teacher-scholar, she has mentored over 100 research students, the majority of whom continue to work in science and health. So thank you very much for joining us. Professor Okeke, please take it away.
Okay, good morning, afternoon, evening, depending on where you are. And Benet, thank you for the introduction. And also thank all of you for the invitation. It's really nice to be here. And I suppose one of the real pluses of the pandemic, one of the few pluses, I should say, of the pandemic is that, you know, I can give a seminar and not have to arrange childcare for travel. So thank you so much for having me. So what I'm going to share with you today is the work that we've done in Nigeria to set up surveillance for antimicrobial resistance, and how genomic surveillance is a really crucial part of this, how without genomic surveillance, we wouldn't have a surveillance program. And that with it, we have one that is actually, although it's relatively new and still being built, it's actually much richer than we could have predicted. So I should say that I'm really describing a huge collaborative effort between my group at Ibadan, the National Center for Disease Control, which coordinates AMR surveillance, and numerous collaborators, particularly those that are part of the Global Health Research Unit for Genomic Surveillance of Antimicrobial Resistance. So the story of how Nigeria got an antimicrobial resistance surveillance system is part myth, part miracle. This is something that Nigeria should have had for several years and just didn't. And so we're just incredibly proud that we actually have one, even though it's small and it has its limitations. And so I wanted to spend the first part of the talk talking about how we got to that place. And I'm essentially telling a story that Chikwe Ihekwazu, who's the director of our National Center for Disease Control, has told in a thread on Twitter 2 years ago. So the story actually starts before any activity. It starts in a place where there were numerous, well, not numerous, but quite a few academics, myself included, who studied antimicrobial resistance in Nigeria, but there was no contact in the Ministry of Health or in the Ministry of Agriculture, or in the Ministry of the Environment. And these are the three ministries that really should be concerned with One Health surveillance of antimicrobial resistance. And AMR was really just not a health priority in Nigeria, as is true in most of the world. There was a lot of untapped expertise in the country, because when interest in planning and acting against antimicrobial resistance came about, many Nigerian researchers, again myself included, were called up by other African countries to help them develop national action plans. And we did, but we just didn't have one in our country. Now, in 2014, as those of you who do antimicrobial resistance research will know, the WHO published a global report on surveillance of antimicrobial resistance. And this is the key figure from that report as pertains to Nigeria. And just to tell you, if you don't want to bother reading the legends, that the greens here are not places with resistance. They are places with data. And the lighter green you are, the less data you have. And Nigeria, which hopefully you remember is in this corner of West Africa, as you can see, is completely white, as are many other countries in Africa. One of the things that WHO highlighted in that report was that there were many parts of the world, particularly in Africa, for which there was no surveillance data that could be used for burden calculations or for informing interventions. So at the 2015 World Health Assembly, the WHO presented a global action plan on antimicrobial resistance, and then actually asked all member states to have in place 2 years later national action plans that spelled out how they were going to address antimicrobial resistance. And Nigeria, being a very prominent and loud member of the World Health Assembly, of course, signed on to this task, and everybody went home from Geneva. Now that was May 2015. In August 2016, this is how much activity that Nigeria had seen in terms of developing a national action plan. Contrast it to Ghana, that actually had a plan by 2015, but was honing it. Nigeria had no plan and no activity. Now, towards the end of 2016, Chikwe Ihekwazu was appointed the director of the National Center for Disease Control. It was a really interesting year. The end of 2016, that was the same year that Sani Aliyu was appointed the director of the AIDS Institute. And 3 years later, for the first time, Nigeria actually had an estimate of how many people in the country were HIV positive. So we had this, flurry of competent and driven people, great leaders who were appointed to head important health agencies. And my colleague and I, Dipo Abodering, were talking about the list when we saw the list come out. And we said, we should reach out to Chikwe and remind him, if he doesn't already know, that this antimicrobial resistance thing is going to come and bite us in less than a year's time. So we met with Chikwe in December 2016. We told him what the mandate was, and he wrote to the Minister of Health and asked for permission for NCDC to pick this task up and run with it. I'll spare you the details as we only have 45 minutes, but essentially we worked like crazy for 3/4 of a year. And by the end of that time, we, and this is a very royal we, because it includes stakeholders from all the relevant agencies. It includes people from the private sector, it includes the NGO sector, because in the end, we actually all knew these people. We just never got ourselves together in the same room. We were able to do a situation analysis of AMR in Nigeria, which is normally a prerequisite for writing a plan, and write a national action plan for antimicrobial resistance. And right after the 2017 World Health Assembly, at which Nigeria did actually submit this plan, WHO published a world map, but I've just cropped it around Africa so that you can see, that showed progress towards national action plans. And in this map, if you're red, there's really no progress. If you haven't really got a color, then nobody's sure what's happening in 2017. And if you're green, you actually have a full multi-sectorial plan in which all agencies that are responsible have signed on. And as you can see, there are not very many greens. And most of the greens had some surveillance data in 2014. If you're dark green, it means that you have a plan and you actually know how you're going to pay for it. And South Africa was the only country in Africa that was actually at that point. So Nigeria, our colors being green, white, green, we were very proud in this instance to be green rather than white. We had actually exploited the fact that Nigerians are easily embarrassed when shamed. And we had said, look, we had to do this by this deadline, and we'd actually made it work. It was a really local effort. We did use external resources. We got small grants from CDEP and from REACT. But in terms of the actual work that was done to pull together the plan, almost all of it was done in country, and it was done by local stakeholders who now have ownership of the plan, which was really important to those of us who coordinated the effort. Of course, when we had produced this, on the one hand, we had done the task by the deadline. But what this meant was that there were really great expectations. You don't just have a plan, you have to do something with it. And so the next step, obviously, was to implement this plan and stay green on the map, if you like. So one of the things that was in our plan, our second pillar, as it is in all national action plans that are modeled on the global action plan, is that we had to have a surveillance system. We had to have a system that would help us measure what the antimicrobial resistance problem was, and also then be a tool that we could use to assess the impact of interventions when we implemented them. Now, while we were doing our situation analysis, we rapidly realized that a lot of people were measuring zone, well, I shouldn't say measuring, were making zone diameters. In fact, a lot of them were actually not measuring the zone diameters they were making. But we had enormous issues with how antimicrobial susceptibility testing was being done in many laboratories. And this made a lot of the work that was ongoing unsuitable for a surveillance system. So if you don't have a surveillance system, you basically can't see. And not being able to see means that you don't know when you're making progress, and in many instances, you can't make progress. We decided that our best bet was to organize our surveillance system around the guidelines provided by the global antimicrobial surveillance system that is coordinated by WHO. It's known as GLASS. And so to do that, we needed minimally components that included a national coordinating center, which would be our National Center for Disease Control, NCDC. We needed at least one sentinel site. I mean, GLASS is great. It's written so that a country with minimal resources and a lot of drive can start the process of building a surveillance system. So you need at least one surveillance lab, and you need a national reference lab. So the first thing that we had to do when we decided that we wanted a surveillance program was to get these components in place. And we actually signed on to GLASS early. There was an early sign-on, which also closed in 2017. We signed on early not because we had the components ready to go, but because low and middle income countries that signed on early would be able to access technical support from WHO. And we knew that we needed that support to build a surveillance system. Now, some of the issues that we had to deal with is that it's not enough to just pour agar, put organisms on it, and drop discs. You've got to have a quality management system in order to generate data that can be used for patient care and for surveillance. And the trouble is that these quality parameters are actually very, very uncommon in Africa. The map on this slide shows the number of labs that were accredited in 2017 across the country. This is the number of all, across the continent. This is the number of all labs. So it includes the HIV and TB labs, which form the majority of labs that were accredited across the continent. Most labs are working without one or more essential quality components. And we had to sit down and decide which quality components we could do without and be okay, and which ones which we absolutely had to have. And then we built a tool for assessing labs. So our object was to build a surveillance system similar to what, for example, the Philippines has. Celia Carlos in the Philippines has built this amazing surveillance system that is representative, that produces high quality data and really gives them a really nice picture of what's going on in terms of antimicrobial resistance. The trouble is that it took Celia 30 years to build this system. I know because I was at the 30-year birthday bash. And I remember sitting there thinking, this is fantastic, but we don't have 30 years. And I wasn't the only one. Onyi, one of our technical working group members at the time, tweeted, you know, how can we get this? We need this now, now, now. What are we going to do? The good news is that we don't have to wait 30 years. As Kwame Nkrumah famously said, we can use knowledge and experience that are already available to us by those who actually built things from scratch, did things, made mistakes. We have no reason to make their mistakes. We can achieve in a generation what has taken 100 years. And so this is what we set out to do. And we wanted a surveillance system that, in addition to telling us what was resistant now and how things were changing over time, wanted a system that would be sensitive enough to find new things. For example, you know, half a decade ago in China, a new colistin resistance gene, MCR1, was found in China. And this gene, of course, it took some molecular biology to locate the gene. But the important thing that the first clue that they had this problem and this gene was circulating came from a routine surveillance project. And so we wanted a surveillance system that would be sensitive enough to be able to do that sort of thing. Now, could we get that? Because in the end, what we were struggling to do is to build a set of labs that were testing with disk testing, which is the most cost-effective mechanism that diagnostic labs in Nigeria can actually use. The answer is yes. We have to do rigorous quality assurance of data. We need to make sure that the data that we collect is at least semi-quantitative. And so one of the problems we had is that many labs were just reporting R for resistant, S for sensitive. We would actually need numeric data, which you can get from disk testing if you measure your zone diameters. We needed to look at patterns and profiles as well as individual prevalences. So we needed to handle our data electronically. And we would essentially be able to leapfrog some of the difficulties that other systems had in order to have surveillance systems that were reliable and sensitive by including genomic surveillance from the start. And actually at this point, it's pretty obvious that that was the thing to do. But in 2017, we actually got a lot of kickback on this. A lot of people said, you don't have any money. Why do you want to introduce genomics at the start? We had good reasons. We laid them out in the plan, and then we went to find some money. It was really, really key for us to have this. And one of the things I would hope that I can do in this talk is actually to show you how important the genomic surveillance component of this is. So we started out by knowing that we needed at least one Sentinel lab, and we needed a national reference lab. And the fastest way to find out who was either ready or almost ready was to make it voluntary to apply to the surveillance system. So we sent out a lot of calls. We sent out a tool that we had developed that would allow labs to self-assess. And then we asked labs to return the form. And when they returned the form, we took a look at the results. The results were not good. The first batch, I mean, there must be, say, about 1,000 or more labs in Nigeria that are doing susceptibility testing. Our first batch, we had less than 30 labs respond. So again, not a lot of people were volunteering to jump into the surveillance system. And this is a problem that we face until this day. Our current system now consists of 11 labs, and it's still heavily weighted towards the southwest part of the country, which is where you have more resources for medical lab science. So I'll spare you the gory details, but I did want to just show you some of the things we found in the lab. So we had a simple traffic light system. If you were red, you were not ready for the surveillance system yet, and we'd have to do some major work with you to get you ready. If you were yellow, you weren't ready for the surveillance system yet, but with some minor work, and if you worked hard, we could get you there. If you were green, you still weren't ready for the surveillance system. Originally, green was supposed to be ready, but then we found no lab was ready. But if you were green, we could do a few things and you would be ready. So essentially, in our first batch, we only had 2 green labs. This was a problem. One of the things you'll notice from this slide is that almost no lab was doing external quality assurance. And this is not because they were sloppy or evading quality. It was simply, as we found when we tried to procure quality assurance panels for the labs, that it's really difficult to get them into Nigeria. Nobody makes them in country. With the red labs, we saw very commonly the labs didn't even have quality control organisms. Many of them were not using the right technique. Very few of the labs even had SOPs, so how could they use the right technique? And there were a number of infrastructural shortfalls, including the fact that many of the labs didn't even have access to purified water. Now, the yellow and green labs, we didn't have that level of problems, but we did have issues that had to be resolved. So what we decided to do with the first batch and the second batch, we pulled them together, and in the end, we ended up with 5 yellow and 2 green labs. We decided to start working with these 7 labs. The other thing we had to do is that although the National Center for Disease Control does have labs, at the time they had no bacteriology. So they could not serve as a national reference lab. So we had to find a national reference lab. And so what we did was just to say, we have to pick one of the green labs and rapidly turn it into a national reference lab. So no surprises, our first year enrolled in GLASS, we didn't actually submit any data. We got to work. We did a lot of work communicating with the administrations of the hospitals so that they would provide support to their labs and deal with the minor infrastructural deficiencies where those were present. We trained and we trained and we trained. And although I'm seeing myself in a lot of these pictures, it wasn't just me traveling around to every lab. We had a huge team of volunteers who helped. Not all those volunteers were from within Nigeria. You can see up here at the top, this is a group, Mary Chataway and Colin Brown, who came from Public Health England to help us do some of the training. The labs were also really hardworking and really flexible with us, willing to subject themselves, if you like, to this rapid training so that we could essentially bring our quality up to scratch, or at least up to the minimum levels that we would like. And by our second year enrolled in GLASS, we were able to submit data from 3 participating Sentinel labs. So we had a national reference lab and 3 labs. And then we've continued to grow it from there. So let me return to this question of genomic surveillance, which is where we'll dwell for the rest of the talk. Why did we want genomic surveillance? Why did we think that we absolutely had to have genomic surveillance in Nigeria, even though it was costly? First of all, we needed reference lab services. You remember that we picked a reference lab out of one of the 2 labs that was most ready, but not quite ready to serve as a Sentinel lab. So we needed to be sure that the reference lab functions could be provided within the surveillance system. We decided that it would be easier to build genomic surveillance into the system than to build in phage typing, serotyping, what Mark Ackman would refer to as YATMs, yet another typing method. So it would be simpler to build in one method that would do all these things, rather than to build in a lot of different methods, which quite frankly are on the way out. We wanted to make sure that NCDC, which is essentially, NCDC is our CDC, by the way, they have a lot of responsibilities, including, for example, managing the COVID response. So we wanted to make sure that what we built would be useful to them in other ways, you know, and so having genomic surveillance would certainly be useful in that regard. We wanted to make sure that when we saw things in Nigeria, we could easily compare them with things that were being seen in other parts of Nigeria or in the rest of the world. And I just hinted to you how difficult it is to get live bacteria into the country for quality assurance purposes. We don't want to be sending strains around. It's so much easier to send genomes around. And then many of us who were helping NCDC with this were academics, like myself. We have this tendency to get bored. And so it was important for us to set up a surveillance system that would allow us to ask and answer questions. And then that would keep us engaged. So we had ambitions to set this up really quickly. We were fortunate to work with David Anderson and others and get funding from the UK NIHR to set up the genomic component of this. But it took us a really long, I said took, it's present tense. It's taking us a really long time to set this up. And it's not the initial things that we're warned about that are the issue. Somebody said, how can you do genomics in Nigeria? You haven't got enough bioinformaticians. This was absolutely true. How are you going to operate sequencers and bioanalyzers and so on when you're having trouble keeping your PCR machines on? So all of these were concerns, but they were things that we could think about and problems that we could solve. Instead, we had other problems, some of which we didn't predict ahead of time. It costs over $100,000 to buy a MiSeq to sequence under Illumina technology. At the University of Ibadan, any purchase of 100,000 naira and above, which is slightly less than $300, has to be signed off by the vice chancellor. So making big, expensive purchases is actually not that easy. And so just as we're revolutionizing how we do science and how we do surveillance, we also have to revolutionize our very old, antiquated administrative systems and make sure that they're agile enough to ensure that we can do genomic surveillance. Consumables are really difficult to get hold of in Nigeria. We pay more, we get less technical support, we get less service. So these are things that we're still battling with now, not just for genomic surveillance, but for anything lab in Nigeria. It's hugely complicated to be able to do things that require all of these consumables, none of which is made locally, and which have to be imported through supply chains that are actually very weak. I've talked about EQA, so I won't go there anymore. And then, you know, when we started off, we said, well, there are all these tools in the cloud that we can easily use, that our bioinformaticians can use very easily, and they were trained to use those tools. And as soon as we got our first batch of genomes, we realized that we were stuck. I mean, I was telling Benet just before this meeting that I'm hoping that I can stay online long enough for the talk because our internet is so bad. So how are we going to do sequence analysis in the cloud if we don't have the bandwidth? So these were, again, things that we had to look at. They all have solutions and we're working through them. But these were problems that we didn't immediately envisage might come in the way of successful genomic surveillance. So if you ever visit Ibadan, and you should, what you'll find is that we actually have a bus stop called Challenge. Challenge is not new to us. Okay, as I often tell my students, we stop there to make connections, but we don't stop there for very long. Okay, so we have challenges, we figure them out, and then we move on. So one of the root causes of why people are apprehensive of deploying genomic methods in African countries is that there was a period when everybody in the world was learning and doing molecular biology, and Nigeria was battling with a structural adjustment program. Our curricula were not updated. Your average student who has a degree in the biological sciences has done very little molecular biology. So being unable to understand the biology and, by the way, the computer science that is needed to do bioinformatics, being able to make that loop is a bit challenging. And so we're doing a lot of groundwork with curricular updating so that in the future, being able to train bioinformaticians, to train laboratory scientists to do sequencing will be a lot easier. The other thing we're doing is working with friends. These are not just our problems. They're problems that many lower middle income countries have, particularly those in Africa. And we're fortunate to be part of 2 consortia that are building capacity for genomic surveillance of antimicrobial resistance. The first of which we've been members for a year is the Global Health Research Unit that's funded by the NIHR. And this is a 5-way collaboration that's allowed us to develop templates, toolkits, protocols, and so on in the group, and then tailor them for our individual sites. We wrote a supplement to our grants to focus on good financial grant practice so that we can deal with those administrative challenges that I mentioned earlier. We've developed 4 different models for setting up sequencing if there's no pre-existing equipment, depending on what kind of support and so on that you have. So these are models that actually other countries could deploy and hopefully not have to use the equivalent of 30 years, more like 30 months that we did to be able to build up our genomic surveillance. And then we set up a lot of pipelines, which means that although I have 2 very highly skilled bioinformaticians, most of my group can do the operations that they need to do to be able to figure out what's going on with antimicrobial resistance. And all of us have integrated what we're doing with national and international surveillance systems. We're working through NCDC, and NCDC submits to GLASS. So with the time we have left, I want to show you some examples of how valuable genomic surveillance has been to us. And the list you can see up here are some of the areas that before we started, we figured out that genomic surveillance would offer us extra benefits. These were some of the things that we used to justify introducing what was then considered expensive technology. But interestingly, it just costs you a little bit more to sequence than to use a Vitek machine in African countries to get a biochemical profile and an antimicrobial resistance profile. So I think expense is really not a huge argument anymore. So I'm going to share with you today just an example of how genomics sequencing has allowed us to provide those essential reference lab services. And then I will also show you examples of how genomic surveillance is helping us to find outbreaks of resistant bacteria. So in the GLASS manual, which every body building these systems would have read cover to cover several times, these are the functions that a national reference lab is supposed to have. Okay, they include training, obviously, which you've seen that we're doing, but also doing some primary analysis of samples where necessary and confirmatory testing that occurs at sentinel sites, doing subtyping, doing some research, helping with quality assurance, and doing outbreak investigation. So you remember that when we did our lab assessments, even the labs that were green, that had pretty good 2018 EQA results, they had trouble with strain subtyping. This is an area that you need a lot of carefully honed expertise in many different areas. And so we didn't actually have a lab that was going to be able to subtype strains below the level of species. And you need that if you have a national reference lab, because you need to understand which clones are circulating in your country as well as in different institutions. Also, we didn't have a lab that had an identification process that was strong enough to be able to do confirmatory testing. After we had sequenced our first batch of isolates from the labs, we found out that many of them have a species, Enterobacter holmachii, that was never on the radar in Nigeria before we started genomic surveillance. There's no lab that submitted a strain to the National Reference Lab and said, hey, I have Enterobacter holmachii. A few labs in the second column here told us that they had Enterobacter cloacae, which is okay because Enterobacter holmachii is part of the Enterobacter cloacae complex. However, even Vitek, which is sort of recommended by GLASS as your sort of minimum, not necessarily Vitek, but any automated biochemical testing system, could not identify Enterobacter holmachii. It identified the strains as the complex, which again is okay, except when you find that within our Enterobacter holmachii strains, we have certain clones that are overrepresented. And so they are clones that we really should be tracking. And so if we didn't have genomic surveillance, we would need something at least as good as the Vitek machine that we have, as well as something that would subtype these strains further in order to understand what was circulating in our environment. And then we would obviously then need to feed back to the labs what we were getting and help them get to a point where, at the very least, they're making a diagnosis, an identification of Enterobacter cloacae. Okay. Just to say off the side that although the primary aim here is to confirm the identifications that the labs are making, we get a whole genome when we try and do that. And so with that whole genome, we can tell the labs which resistant genes their strains have, and we can compare this to the resistance profiles that they've generated. We can tell the labs which strains are likely to have plasmids. We can tell the labs which strains are actually in clusters that look like they might be from outbreaks and ask them to look at the temporal history of these strains and confirm whether or not these are outbreaks. So these are add-ons that we can do just because we wanted to confirm the identity of strains using genomic surveillance instead of using one of the alternative methods that would have been available to us. Sorry, I'm just looking at the time. Okay. So in terms of hospital outbreaks, if we have time, I'll share two examples, but at the very least, I'll share one. So one of our Sentinel labs had a great clinical microbiology resident, Emmanuel Irek, who was actually really careful. First of all, he noticed that a lot of the Acinetobacter strains in the lab were being misidentified as Staphylococcus. This may sound strange to you if you know that Acinetobacter is gram-negative and Staphylococcus is gram-positive. But it turns out that if you don't do your decolorization right, you end up with something that looks like a gram-positive, very short rod, which could look like a coccus. And then you mistake it for a completely different species. So he discovered this was happening. He intervened. He was able to get his Acinetobacter identified. And he told us that he suspected that in his ICU, he had an outbreak. So he sent us all the Acinetobacter isolates that they had from the facility. We got a few more from a few other facilities. We sequenced them, and lo and behold, he's right. The A cluster here is an outbreak of strains from his ICU. It's a carriage outbreak, which is what he thought he had. And a few of these strains have also caused bloodstream infections. The interesting thing is that he just sent us a bundle of Acinetobacters that he'd got from the whole hospital. And we found in there another cluster of strains that have the same resistance genes and very few SNPs between them. So it's highly likely that what was going on, that he had an outbreak that he wasn't aware of, as well as an outbreak that he was aware of. And this is the sort of same thing that we're seeing with pathogen after pathogen as we use genomic surveillance methods. Currently, we're a little slow because we're still learning our genomics. So currently, and unfortunately, I should say, we're identifying these outbreaks retrospectively. But going forward, and certainly by later this year, we should be able to identify things in more of a real-time mode. And we're hoping that this will help support infection control committees in the institutions that we work with. In the interest of time, I'm going to shorten what I was going to tell you about Klebsiella. But I do want to mention that there's a huge burden from resistant Klebsiella in developing countries that is growing. And in Nigeria, we don't yet have a lot of carbapenemase producers, but we do have extended spectrum beta-lactamase producers. And carbapenems are really out of reach for routine use. This is a case study, it's a bit old, that showed a child that was sick for over 6 weeks in a teaching hospital. She'd been referred from a secondary care center, so she'd been sick there for a while. And the amount of money that was spent keeping her alive until susceptibility testing was done, and it was found that what she had was an ESBL organism. She was given imipenem, and she went home happier if her family was a little poorer. And just to say, $500 may not sound like a large amount, but in the time when this happened, this would have been the annual income of her family. So we really can't afford the costs that resistance brings, even when those costs may look small. Whereas look at the cost of the laboratory investigations. They were expensive, almost $20, but when you consider what they would have saved, they're certainly cost effective. So as I mentioned, our genomic surveillance is revealing to us that we actually don't have a lot of carbapenem resistance, but we have tons of ESBLs in our Klebsiella. Most of them are CTXM15, which was the same kind of ESBL that was found in that little girl that I just described to you, and that we find them at all the facilities. When we look at the isolates that we get from each facility and look at how diverse are they by SNPs, by MLST, or by K-type or O-type, one of the things that we found was that the institution that was sending us the most Klebsiellas had very little in-institution diversity compared to other institutions. And when we sequenced the organisms, lo and behold, we found out that they have had a lot of retrospective outbreaks. Okay, these are just two examples. And these two are shown to you because they were actually temporarily clustered as well. But because some of the isolates were misidentified, the lab didn't get a flag that they had many Klebsiella isolates. What they thought they had was 3 Klebsiellas, 2 Enterobacters, and so on. So really building quality at the Sentinel lab site is something that we're still working on. And interestingly, some of these outbreaks are caused by multiply resistant organisms, like you can see in the lower cluster, and a few are caused by organisms that aren't resistant to many antimicrobials. And so if we have the advanced lab information, they actually would not be hard for the infection control committees to search and destroy. So what are we doing now? We are growing our network, continuing to focus on quality. And we're actually thinking very carefully about how to do genomic surveillance below the level of tertiary care. And I want to explain why we're doing this. So a paper was published in 2018 from a network known as ANISA that looked at a number of institutions across South Asia. And what they found that was quite interesting was that they were finding most of the isolates from these small hospitals were actually susceptible to cheap antimicrobials. Now, we presume that we have a lot of resistance because all of our surveillance is done with tertiary care hospitals. And as you can see, we're struggling with building quality in diagnostic labs in tertiary care hospitals. So the idea that we would be able to do this at the secondary care level is certainly possible. We are working on it. NCDC actually has a batch of secondary care labs that we're now going to be working with to build quality bacteriology testing. But it's going to be challenging to have a good range of non-tertiary care hospitals in our surveillance network. And as you know, all around the world, difficult cases are referred to tertiary care centers. So if you're only doing your surveillance in tertiary care, you may have a bias towards only looking at resistant strains. So we're really asking the question of how do you do surveillance below the tertiary care level? And in fact, not just at a secondary care hospital, but a rural clinic that has a small lab. Could they be part of the surveillance efforts? And so to do this, we're going to have to think of different methods. We are not going to put a MiSeq in a rural lab because we're struggling to have one at a university research lab. But we could perhaps use nanopore sequencing at primary and secondary care levels if we got the parameters right. And so that's something that we're piloting right now. Different ways that we can use nanopore. Can we enrich for certain pathogens in specimens so that we might be able to even do direct from specimen sequencing? If not, at the very least, can we isolate the most common organisms and sequence them in a rural hospital? This is something that we're testing out now, and hopefully in a few months, I'll be able to tell you. Yeah, I'd like to end at this point because I do want to leave time for questions. I really want to thank my team. It's been a struggle, but a really exciting struggle. Most of them came into my lab as master students and have essentially learned a completely new set of skills very, very quickly so that we can roll this out across the country. I'd also like to thank all of the individuals that we've worked with, as well as our sources of funding. Our genomic surveillance for our national surveillance with genomics is funded by the UK National Institute for Health Research. We are part of another consortium that does genomic surveillance for West Africa called Seek Africa, that's funded by the Fleming Fund. And our piloting ideas, the risky project, if you like, piloting ideas that could be used below the tertiary care level is funded by the Bill and Melinda Gates Foundation. In addition to our group, I have really enjoyed working with individuals at our Sentinel Labs, at the National Center for Disease Control, the Technical Working Group for Antimicrobial Resistance that led the effort to write our plan, as well as colleagues at Public Health England and the Wellcome Trust Sanger Institute, which has been sort of our school for sequencing. So I understand there'll be questions now. I think I've gone a little bit over. So if I don't get to your question, or worse, if I do get to your question and answer it badly, my email address is in the corner of the slide, and we can pick up the discussion after the talk. So thank you for coming. Thank you for your attention. And I'm really interested to hear your questions. Thank you very much.
Thank you very much for this very terrific talk, very interesting. And thank you also for being mindful of the time, Professor Okeke. It's really appreciated. So we'll take questions now. And actually, we already have questions from the audience. The first one is from Bolu Fakoya. Is there any role for private partnerships in procuring technology for this genomic analysis?
Yes. So in many, not just in procurement, but in many other areas. But I think let's dwell on procurement first, because one of the models in African systems is if you're doing work in a university, which is in a university like University of Ibadan, if you're doing work in a university like University of Ibadan, the administration is built on 1930s British bureaucracies. It hasn't changed since then. It's not agile enough to do anything. And so what people normally do is they set up an NGO, they put their grants in the NGO, it's administered really well by the NGO, and life continues. But the problem with that is that if my student gets a fellowship, that isn't going to let them set up an NGO. It has to be administered within the university system. So we are rather bravely, I would say, we are trying to see whether we can build administration for procurement, for financial management, and so on within the public system. Now we well may fail, but I would really like to say that I tried before that happens. Now another lab in Nigeria, which many of you may be familiar with, that does genomic surveillance, but largely for viruses and also malaria and other things, is based at a private institution. And they have some of the problems that we have. They're based at Redeemer University. PI is Christian Happy. They have some of the problems that we have, but not as many. And so, yeah, in the private sector, things are better. But the private sector in Nigeria is really small. And so we'd like to see how well we can work in the public sector. And I think NCDC is a model of that because it's a Ministry of Health agency and it's really efficient. And so I think if we try a bit hard, we might be able to do that. In terms of partnering with private sector in general, yes, I think we absolutely have to do that. As I mentioned before, our public sector is not efficient. It's also poor. There's a lot more funds in the private sector. And so we really do need to work with private institutions. We're interested in working with private labs, but we're holding back a little bit because right now what we have access to is funds that can be spent in public labs. And so we will fold in the private sector. The only accredited bacteriology lab in the country is a private lab. So we will certainly fold them in, but that is going to be in parallel with the streams that we're working on right now. Thanks for the question.
Thank you for your answer, Professor Okeke. We have a question from Amy Taylor. Are capacity building efforts coupled with other types of genomic surveillance capacity building? So for example, could we use the surveillance network to survey, like to keep an eye on other diseases like malaria, for example?
Yes, this has always been our aim. Now, the good news is that we're not the first to try genomic surveillance in Nigeria. So I mentioned Christian Happy's group, for example, started off with virology, is broadening, is much bigger than us. And so, you know, when COVID came along and we needed to do genomic surveillance for COVID, it made sense for Christian to pick this up immediately. So yes, our facility right now is devoted to AMR, but is open to receiving cholera, for example. We're set up, trained, and functional for cholera, which is a bacterium. And eventually we will expand into other things. So the whole idea of picking genomic surveillance is that it has this flexibility, allows you to do other things. And we, as COVID has shown us, we don't know what the next thing will actually be. So we need to be ready for it.
Thank you for your answer. The next question is from James Otieno. Could you talk about turf wars that often happen in country between labs or between government technocrats and scientists that end up hampering progress in setting up surveillance programs?
Sorry, could I talk about what was the word?
Turf wars.
Turf wars. Ah, yes, there are always turf wars. The interesting thing with genomic surveillance is that right now there's no turf. So there's nothing to fight over. We need to be generating 100 times as much sequence as we're generating now. So if more people want to come into the space, please, by all means. So I think right now we don't have issues with turf wars because there are too few of us doing it. But it's going to happen in the future. And I think when it does, for me, I have a whole research program that's so cool. So if there are so many people who want to do genomic surveillance, I was asked the question of, is it a good thing that all our genomic surveillance is in academia? And I said, no, it's not. And I don't plan to have this in the next 10 years. I'm hoping that in the next 10 years, NCDC will take this and own this. And maybe, you know, they'll call me once a year and just tell me how things are going. So once it gets to the turf war level, I'm sure it's going to be bitter battles, but I assure you I won't be part of them.
All right, makes sense. The next question is from Magic Senghor. So he thanks you for your really inspiring work. And his question is, regarding the retrospective outbreaks, is there a systematic framework for selecting data sets that would allow you to reconstruct the evolution of nosocomial outbreaks over time?
Yes. And now I'm wishing I have Ayo. Ayo's my lead bioinformatician. I'm wishing I have him here, but he's going to have to watch me mess this up and then tell me what I should have said. So a few things that we do. First of all, we ask the lab to go back and get us temporal data. Can they show us temporal and spatial, actually? Can they show us these isolates that they sent us? When precisely were they isolated and where were they from? If they were all in January 2021 in the ICU, that's our first clue, even without looking at the genomics. Now, Ayo actually has a pipeline that he built with Sylvia Argumon and Anthony Underwood at Sanger that actually helps to look at SNP patterns, because we found that for some labs, they miss out some piece of information. They're not sure what the date was for this isolate. So we can actually look at the genomic data and figure out, based on number of SNPs, is it unlikely by chance that these two things came together at the same time? Essentially, that's the big picture question. And if the answer is it is unlikely, then it's probably an outbreak. And the nice thing that Ayo did was that he ran this, and then we also looked at the temporal and spatial data. And so we had confidence because he was finding the same outbreaks that the space and time data were finding. So yes, we have genomic ways of looking at it, and we encourage the labs to look at it, because what it's doing is that it's strengthening their infection control teams. And then that would make it more likely that they would find something that they suspect is an outbreak, send it to the reference lab as an emergency sequence batch of isolates. And then we can return data in time for them to actually be able to do something about it. Thanks for the question.
Thank you for your answer, Professor KK. Mark Lipsitch would like to ask his question. Mark, could you, yeah, he's here.
Thank you. Thank you, Aruka, for a great talk and for especially the inspiring vision, which I didn't know came from Kwame Nkrumah, about lack of legacy being a spur to ambition, but also showing that that can really work in practice. That was incredibly exciting. My question is about the old problem of how to get representative samples, and just curious how you think about it. I mean, it's all repeating itself in COVID, that we both want to see the most interesting stuff, which means doing very strange types of sampling, and also to track frequencies, which means doing very representative sampling, and both of those are hard. So I'm curious if you could just talk a little bit about how you've thought through those problems.
Yeah, it's a really important question. Thanks, Mark. So right now, nothing that comes out of Nigeria, nothing that I showed you is representative of anything. Okay, so right now, most of our 11 labs, I think something like 6 of them are in the southwest of the country. None of them is representative of the state that it's in. So we didn't have originally we actually had a sampling strategy that we divided the country into states and regions and so on, and we're going to take one hospital from each. And we just realized that, look, in some of these places, there's nothing in the hospital. So saying that this hospital or one near it would be part of our surveillance system would mean that it would take us 30 years, essentially Celia's time, to be able to build the system. So what we did at the hospital level was we had asked hospitals to volunteer. And of course, we got volunteers from the strongest hospitals, most of which are within commuting distance of Ibadan. So even in terms of who's part of our surveillance system, we're completely non-representative. But what we're hoping we do is that we build enough capacity in these institutions so that they can help spread the word and train other people. And then somewhere down the line, we can actually look for representativeness in terms of who is part of the surveillance system. That's the first thing. Now, within a particular hospital, the isolates that they are looking at are not select, or let's even start with the specimens. The specimens are not selected in any representative way. In fact, I'll horrify you by telling you that if a child has a fever and is likely to have bacteremia, whether or not they get a blood culture depends on what the parents can pay. So blood culture bottles are the most expensive things that our diagnostic labs buy. They don't have any money. There are a few labs that we've been able to support by giving them blood culture bottles so that they can do free blood cultures. But we can't do this nationwide. We need a strategy. I am arguing that blood culture should become the purview of the institution and not the patient, because in the end, by the time we have the blood culture result, most patients are either almost better or dead. It's the institution that benefits from a blood culture. So we have very few blood cultures. In fact, some hospitals we're finding we're only doing 3 blood cultures a year because only 3 patients could afford to pay for blood culture. So this is a huge problem. And then when you get the isolates that you get, originally we had said, well, they will be sending 1 in 10 to the reference lab. But the fact of the matter is that none of our Sentinel labs is getting that many isolates. So right now they're sending everything to us. And as at last year, we were still sequencing everything. The numbers are that small. And when you consider that Nigeria is the most populous country in Nigeria, as I mentioned before, nothing is representative of anything. So we have in our National Action Plan some loose strategies of how institutions were supposed to be selected and how strains sent to the reference labs were supposed to be selected. But we're not pursuing any of that now just because the numbers that we have are so small. And I'd encourage anybody who looks at our data, either here or subsequently when we publish it, we will put it in the limitations section just to know that this is not representative of anything. It's really just a start. But we hope to grow. You may have heard of the Pathogen Genomics Initiative in Africa. Gates is giving African public health institutions Illumina machines. So things are going to scale up pretty quickly. And when they scale up, that question of how do we get representativeness now that we can actually do it is going to be really, really important to revisit, because I'm not convinced that some of the loose strategies that we laid out are sufficient, particularly towards this problem that I was mentioning of we have to be able to bring in things below the tertiary care level. Otherwise, it's really just a picture of who didn't recover when they had received three courses of antibiotics. So thanks for the question.
Thank you, Mark, and thank you, Professor Okeke. I think for the interest of time, maybe we should stop here. So if you have any questions, feel free to reach out to Professor Okeke directly. On behalf of the Center for Communicable Disease Dynamics and the Harvard T.H. Chan School of Public Health, let's thank Professor Okeke again for joining us today and for her insightful, interesting, and terrific talk. Thank you to everyone in the audience as well for your engagement, for your questions, and please do get in touch with any of the seminar organizers if you have any questions for us or for Professor Okeke. Please check out the CCDD website to stay up to date on upcoming seminars and other events. Thank you again for joining and enjoy the rest of your day.
Thank you for having me. It's been fun.